We're going to go ahead and we will hear from our guest tonight, Rachel Gauza. She is the Principal Natural Resource Specialist with Parks Montgomery, and she's also the Biological Monitoring Program Coordinator. So Rachel, welcome tonight. Great to have you here. Please go
Hi.
ahead.
Yeah, thank you so much for having me. Mike, thanks for that great introduction to biological monitoring. It sets the stage very nicely. I'm pleased to be here today to share a little bit about Montgomery Park's biological monitoring program and the data collection in the Sligo Creek watershed. I'm going to be focusing what I'm sharing with you on one site in particular, which some of you may even recognize as that bridge behind me in the photo. So just a little teaser there. Next slide, please. So our biological monitoring program is part of our resource analysis section within the Park Planning and Stewardship Division, and our focus is on aquatic resources management. So in addition to our biological monitoring program that's helping to kind of keep the pulse on our park's aquatic ecosystems, our resource analysis staff is working together on coordination and fulfillment of all the elements of the NIPD's MS4 and industrial stormwater permits, as well as oversight of restoration and mitigation planning to ensure that there's impervious coverage treatment requirements being met. And the reference for the water watchdogs at the ice rink, we actually had a staff person out there working alongside DEP as well, tracking down the source of that pollutant. So thank you for being our eyes out in the field when we're behind our desks, because then we can join you and make change happen. All right, next slide, please. So our biological monitoring program relies on having 75-meter stream in length stream stations that are basically set up throughout our park system. We work cooperatively with Montgomery County Department of Environmental Protection, Maryland Department of Natural Resources, and others to assess stream conditions. So we also are evaluating spring benthic macroinvertebrate communities. We're doing it just the one time. And we have a slightly different process using a DNET, like the Maryland Biological Stream Survey, like Montgomery County DEP, and then processing our benthic macroinvertebrates in the lab to genus level. So a little bit more specific than what you're able to do stream side, although I admire the ability to be able to identify benthic macroinvertebrates in the field stream side to family level. It's better than I can do. In the summer, we turn our attention to our fish community. We're doing a dual pass electrofishing survey, so limiting our evaluation again within that 75-meter stream station that we are fishing to completion. So 2 passes with an electric pulse that temporarily stuns the fish, and then they are returned after we count and collect our data from them. And then during both those spring and summer monitoring visits, we're also collecting some supplemental data in an effort to get a holistic approach and get a more complete picture of what's going on at the streams. So that includes habitat assessments with a variety of parameters, looking at both the riparian areas adjacent to the stream as well as direct in-stream parameters, physical chemistry measurements, so not the full suite of some of the various chemicals and bacteria that you heard of, rather things like dissolved oxygen, pH, temperature, conductivity, that sort of thing. And then we also are conducting amphibian and reptile searches in those riparian areas, as well as in the stream channel, in order, again, to get that full scope of what's going on at a given time at a given station. Next slide, please. So both the benthic macroinvertebrate and the fish community monitoring does rely on identification and count of organisms, but that isn't really where things stop. Rather, these are examined through several representative metrics that have been tested and validated. And then those are used to calculate a score for each biological community known as an index of biotic integrity score. So you have one for benthic macroinvertebrates, or a BIBI, and one fish index of biotic integrity, or a FIBI. And those can be averaged and combined together for a stream condition score, or can also be examined separately, because each community is responding a little differently to the various environmental changes and pressures that these animals are up against. So with the different scores, we can basically go and say, at this time, according to the benthic macroinvertebrate and fish community, or a combination of the two, the stream condition is excellent, good, fair, or poor. And you can see that breakdown in the little colorful table to the upper right-hand corner. So we're going to take a look now at the fish community composition and scores at a few sites monitored by parks in the Sligo Creek watershed, specifically one of our annual monitoring sites. So let me get you oriented to that. Next slide, please. All right, so what you're seeing is just a subset of all the stations that we monitor. These are the ones we specifically visited in 2020. You'll see we visited 23 stations, 3 of which were in the Sligo Creek watershed. The different colors, obviously the different color polygons there are watershed boundaries, and the different colored dots that you're seeing are different study types. So Montgomery Parks is focusing on targeted monitoring. We're basically putting our resources and our efforts towards where we need additional investigations for planning purposes, where we need to focus making sound land management decisions and make sure that the aquatic resources are preserved within that. And really, in the big part, and probably most relevant to Friends of Sligo Creek, is focusing stormwater control and mitigation in our retrofit efforts so that we can have the greatest impacts in the parklands that need them, basically. So you'll see that we have kind of basically some red dots and some green dots for 2020. The green dots are representing post-restoration stations, and the red dots are annual trend stations that we monitor annually. Next slide, please. All right, so you'll see our red dot now. We're zoomed into the Sligo Creek watershed, and you're seeing our annual trend monitoring station that we do cooperatively as part of a larger Anacostia study with the Metropolitan Washington Council of Governments. It is actually the furthest down county station that we monitor as part of parks. So right before you're hopping over to the PG County border, we get that last little bit of data and information. Some of you may recognize this site as the one near the Carroll Avenue Bridge. It also happens to be a pretty large catchment area contributing to this because it's on the main stem, because again, it's pretty far down and close to where it's going to be joining up with other streams in PG County. You're looking at a fairly substantial drainage area, about 7.4 square miles, which is about 2/3 or so of the entirety of Sligo Creek's drainage. So it's a pretty substantial site and a lot of impervious land cover going to that particular location. Next slide, please. All right, so another colorful chart. Let me go ahead and orient you to it. So this is going to be a plot of our fish index of biotic integrity scores over time. We're expressing the IBI scores, which are from 0 to 5, as a percentage. So you can almost think of it a little bit like a grade. So if the fish community scored a 5, it would be at 100%. Again, those 4 colors are going to represent from the top down: excellent, good, fair, and poor. And so I should note here probably that the data I'm presenting so far tonight is going to be provisional. This is the first that we've kind of looked at it. This is the first we've looked at the 2020 results, like starting about 2 weeks ago. So this is our data crunching season before we go out for next spring sampling. But with that in mind, let's take a look at what we're seeing so far. So the fish IBI scores have been relatively stable and sitting in the fair, or the yellow bar there, stream condition category. You'll see in 2000, the very first data collection event, the stream in this location was poor. And then it's kind of chugged along up and down with some natural variability in the fair category. But in 2020, for the first time in those 15 years of contiguous monitoring, so we're talking from 2005 all the way up to 2020, it crossed over to the threshold in green. So I think we were all a little surprised, and we wanted to know what's going on there, or what might be going on there, and I'm sure you do as well. So let's take a look. Next slide, please. So when we want to understand more about what's going on with a given index of biotic integrity score, a given data point, we start diving in a little deeper to the biological community structure and function for clues. So this is back again to looking at those metrics that I introduced you to earlier. So fish species diversity is going to be one driver behind the IBI and its associated stream condition rating. So the same kind of plot of IBI scores that I shared with you previously up here in white now, and they are overlaid on top of the species count or the taxa richness. So you'll see very generally, like the two plots kind of follow a similar curve with a higher number of species resulting in a higher fish IBI score. Clearly, that's not always the case. As you might anticipate, like in the year 2000, and that was the one that had our lowest score in that category, narrative category of poor, it had relatively few species documented. But then when you go into like 2018 and 2019, you're seeing highs of 15 or so out of the 21 species likely to occur in the watershed. Now, with the caveat that there have been some investigations and studies that say that there's a potential of 70 species that can occur in the Sligo Creek watershed. But I think given the current build out within the catchment area and the watershed overall, 21 is about realistic. And so 15 out of 21 seems pretty promising. But we see also, kind of looking at this trend, oh, well, 2020 looks pretty good, had 14 species collected, but an even higher IBI score. So what's driving that? Next slide, please. And as the next slide, there it comes, pops up. I was going to say, you can probably notice one very glaring exception to kind of the more species, better biological community rule. What was going on in 2015? So this is kind of an outlier. What had occurred a little prior to my starting with parks, so I had to kind of dig around to get the story. But there was a fish kill that occurred within the site a few days prior to the monitoring event. So before the biological monitoring crew went out to collect their data. And when the crew did come out that year, they literally found dead fish. They decided to proceed with the data collection. And what you're seeing on the graph are the only species that had live individuals represented. And I know some of the print is probably quite difficult to see, but what you're seeing is American eel, black-nosed dace, and creek chub. And those are known and documented as being pretty hardy fish. So during whatever happened during this event, either the hardy fish persisted or they were able to basically recolonize or come back into the station following this massive mortality event. So I continued with my detective hat, and I mentioned to you that we look at some physical chemistry, we look at habitat. So I'm looking at that data and I'm like, what is going on? I didn't see anything abnormal for the site. But when I looked at our amphibian and reptile data, I saw that there were also dead larval two-lined salamanders. So another indicator that something had seriously gone wrong there. So as we continued to kind of discuss, and I tracked down the people that were there at the site, we found out that it's unfortunately believed to have been the result of a perfect storm, in the sense that we were looking at a prolonged dry period during the summer months at that time. And when the rain hadn't been falling, that basically, there's some things that go on in the stream that they can already be kind of stressed if it's drought-like conditions or, not having enough rainfall contributing to base flow. But moreover, that you're starting to get pollutants accumulating on impervious surfaces. So nutrient builds up, different chemicals, some of the toxins that were probably referenced in the previous talk are laying in wait, basically. The rainfall comes, especially if it's a very intense storm, and it's basically washing off those impervious surfaces. It's also hitting hot pavement and warming up. And so you could have temperature issues that are coming in and causing a thermal impact to the biological community. So definitely a downer. However, what's encouraging is that if you look in the following years, that the biology basically, at least for the fish community, seemed to bounced back to some degree. It jumped right back up. It was higher than it was in 2014 with different species being there. So what this is suggesting is there is some degree of buffering from the stormwater controls throughout the entirety of the watershed. You know, this poor portion of the stream isn't getting hammered constantly, or you would end up with just 3 species, 3 very tolerant pioneering species. Rather, there is some other protection going on that help offset any potential impacts from more routinely occurring storms. The other thing of note, it would be that it's very possible and likely that because we're so far down in the Sligo Creek watershed, you know, a big main stem station getting close to where it's joining up with other waterways, that there are other populations of fish that can come back in and kind of replace individuals that were lost. So there's at least a robust source population to kind of help in these kind of unfortunate events, and that you are having water quality after that sort of event that would facilitate those individuals coming back in. And, you know, fish are quite mobile compared to something like the benthic and macroinvertebrates. Well, certainly you have adult forms that can fly, but the larvae themselves and nymphs are great indicators because they're staying put. All right, next slide, please. So as this comes up, the other really neat thing that happened in 2016 was not only that species were returning that had been there previously, but we saw a new species that wasn't previously collected at the location. And it wasn't just any species. You can see it highlighted there. And Pat, if you want to pop up the picture of that handsome fella. Let's see if it pops up. If it doesn't, that's okay. There it is. All right. So that little charming creature is a northern hogsucker that happens to be considered pollution sensitive, meaning that it needs a certain water quality threshold to exist in a location. Just as Mike had mentioned that there are certain benthic macroinvertebrates that are sensitive to pollution, here's one of your surrogates for fish. And they rely on large, fast riffle habitat, high levels of dissolved oxygen. And not only were they there in 2016, as you can see what's highlighted, they've been there from 2016 through 2020, you know, suggesting that the water quality is high enough, at least to meet the biological needs of that species for the past 5 years in that portion of Sligo Creek. You know, and of course I didn't forget, and you wouldn't forget, that 2020 also had a good rating for the fish community. But we didn't even collect the highest number of species that year. So what else might be going on? Next slide, please.
Actually, Rachel, just want to ask a quick question because several people have commented on it. They're noticing that in 2020, there's no American eel found. Any comment on that or any thought behind that?
Sure. In that case, it's possible that we just didn't collect them within the 75 meter for one reason or another. They are a migratory species. We do have a sampling window in which from June through September is the only time that we can collect the species so that they fall within the representative protocol and comparable numbers here. But I think that's just a product of natural variability. They're fairly ubiquitous in Montgomery County, but I think that's a great observation and we'll keep an eye on that in 2021. But no, not anything that would dramatically influence the fish community there. All right, so speaking of the fish community there, and this is where I'll kind of start to wrap some of this up and bring some of it back together. Again, these are the metrics we talked about previously. That first metric, total number of species, is just one out of several metrics. So what you're looking at is the 2020 fish community specifically. So just that one data point, and highlighted what basically falls in good, what's falling in marginal or scoring criteria 3, and 1. And, you know, these scores are coming together to get your ultimate rating of good in this case. So there's high number of minnow species, and that's going to be representative of higher stream conditions in Montgomery County. we have higher numbers of fish overall, and with a larger proportion of those being moderately sensitive to pollution, as opposed to just having a bunch of solely tolerant individuals or just a few individuals. That would drive the score down quite a bit. There's also a low proportion of individuals with disease, and that's an important metric when comparing this sample to the reference streams, basically the highest quality streams that were used to develop this mathematical indice. And of course, we have our intolerant species, the northern hogsucker, being supported, which is still kind of a big deal. And you can see how that helped move. If there's an absence, it's already kind of skewing the score to poor. But having one species there bumps the criteria into marginal. And then it would be great if more pollution sensitive organism fish are able to colonize at that point. But there are going to be some holes, as you're saying. So most notably, there is a lack of what we would consider specialist species. So for 2020, just looking at some of these proportions and how the numbers fell out, you're seeing a lot of generalist species. And those are going to be individuals which are able to utilize a bunch of different food sources and a bunch of different habitat types. They're kind of adaptable, if you want to use that term. There was also a notable lack of individuals that feed exclusively on aquatic insects and insects that colonize riffles and those that are really especially sensitive to water quality impairment. So there's definitely a tie in between the benthic macroinvertebrate community and the fish community, which is why we evaluate both. All right, and next slide, please. So I flashed a lot of numbers and colors at you. What does it all mean? Well, good news is there's at least sufficient enough habitat and water quality to support a pollution intolerant fish species, and that's awesome. There are some limitations that are excluding other pollution sensitive species, however, as well as certain habitat specialists from occurring within this reach. And we also, with the caveat, are also thinking about what can we reasonably expect Sligo to achieve. And that's a loaded question, frankly, one that I'm sure all of you and everyone here and then some is still trying to figure out. There are very, very few streams in Montgomery County that have a very high to excellent water quality, and they do look different and are under much different pressures than Sligo. I mean, as friends of Sligo Creek, you know all too well that Sligo Creek has a bit of a history. Obviously, we cannot forget about the benthics. There's a reason, again, that we're looking at these two biological communities. And you can see in the upper right plot there, just a quick look at the benthic IBI scores in red, the combined IBI scores, and then the fish IBI scores again. And the benthic IBI scores are noisy and they're variable. And they are, for the most part, overall lower than the fish scores. So that's another indicator that something is still going on with habitat availability and or water quality that is not allowing the benthic macroinvertebrate community to achieve the same levels and reach their full potential. But results are promising. You'll see the trend lines that are superimposed on there are showing that for both the communities independently and averaged, that the stream condition scores overall are going up. And the 2018 and 2019 benthic IBI scores are comfortably in the fair category. 2020 samples are still being processed, but between the fair category and the increasing kind of trend in the upwards direction, that's encouraging. And, that said, and all that in mind, that we are talking about a very large drainage area, which does make it difficult to tease out both trends and kind of the causes of things like impairment or even causes of improvement. And our biological communities are responding to the overall cumulative effects of these different stressors and changes. So it's really important the work that you all are doing, and especially some of the monitoring that's occurring elsewhere we're not looking at, because we could see, areas in which you're able to draw a pretty good association or conclusion, but it's basically getting watered down a bit at this portion in the drainage area. And it's also possible that any biological uplift is going to be influenced by the neighboring communities. And that's what I mean by proximity to good neighbors. And what I mean specifically by neighbors is not our human neighbors, but the biological neighbors. So I gave you the example of fish being able to move back into the site. Well, how are benthic macroinvertebrates, some of these good ones, even if we restore water quality to a point, going to get from point A into Sligo Creek? So just something to think about, that there can be some real limitations, but that this is a really bright spot in what we've seen in the watershed so far. And that's what I have for you all tonight. If you can put up my thank you slide, I just wanted to acknowledge my team members, because biological monitoring is not a one-person job. Thank everyone for attending and taking the time to listen, and certainly for lending a helping hand to our streams and your aquatic neighbors.
Rachel, thank you very much. Really appreciate you coming by tonight. Very encouraging news too. And thank you for stepping through the details there. It's helpful, I think, for all of us who are interested in the creek and the natural history and the water quality as to how it's all sort of playing together. That's excellent. Thank you.